Good evening, everyone, and welcome to the first of this year's Oxford Botanic Garden and Arboretum Autumn Science Lectures, sponsored by Plants, People, Planet and the New Phytologist Foundation. My name is Simon Hiscock, and I'm Director of the Oxford Botanic Garden and Arboretum. And as usual, it will be my pleasure to introduce tonight's speaker. But first, I have to go through some housekeeping information. First of all, this lecture is being recorded, but your camera and microphone have been switched off, so you are not being recorded. If you would like to ask a question to the speaker, please use the Q&A function at any time during the lecture, and I will ask your question at the end of the talk. If you need to contact us about something technical, then please use the chat function and one of our team will get back to you and try and resolve any issues. The recording will be available to all attendees after the event, and we'll send details of how to access the lecture via the email that you registered with. So now I'm delighted to welcome Dr. Alexander Sandy Hetherington from the University of Edinburgh. It's fair to say that Sandy is a rapidly rising star in the field of paleobotany and the evolution of early land plants. In his relatively short career to date, since his first paper in 2015, Sandy has published 33 papers, mostly in high-impact journals, including Nature, Science, Current Biology, and New Phytologist. He did his first degree in geology at the University of Bristol, which is where I first met him, before moving here to Oxford for a DPhil with Liam Dolan on the evolution of roots. From 2017 to 2020, he was a junior research fellow at Magdalen College just across the road, where he continued his research on the evolution of early land plants. In 2020, he moved to Edinburgh when he was awarded a highly competitive UKRI Future Leaders Fellowship. There, he has built a thriving research team who combine studies of fossil plants with molecular developmental biology in extant plants to better understand the evolution of key innovations such as vascular tissue, leaves, and roots. Earlier this year, in recognition of the impact of his research, Sandy was awarded the Philip Leverhulme Prize by the Botanical Society of Scotland. In his talk this evening, Sandy will explain how UK fossil sites continue to shed new light on the evolution of land plants and terrestrial ecosystems. Welcome, Sandy, and the screen is yours.
Thank you very much, Simon. And thank you everyone for coming. Right, I'll get my screen shared. Bum, bum, bum. Simon, could you just let me know whether that is all looking good?
Looking good, Sandy. All good.
Yep, wonderful. Okay, so thank you so much, Simon, for that lovely introduction. It's such a pleasure to be giving this virtual seminar and kicking off this series on the evolution of land plants. So today I'm going to be telling you how fossil thin sections can really preserve windows into the prehistoric ecosystems of the past. And so to do that, I'm going to tell you first a little bit about what fossil thin sections are and when they were first created. And then I'm going to give you two examples from the research, recent examples from research in my own group, where we're still using fossil thin sections to really begin to understand more information about ecosystems in the past, with one example from the Devonian period and then another example from the Jurassic period. But before we get started, what I want to do is to pose a question for you all. I want you to have a bit of a think yourself about what you picture paleobotanical research could be like, and studying plant evolution. Now, if Hollywood has done its job correctly, I hope at least a few of you are picturing something like this, something like Indiana Jones or Jurassic Park, where we spend most of our time as paleobotanical researchers running into danger, fleeing from danger, and ideally doing this while wearing an excellent hat the entire time. Well, sadly, to disappoint you, is that most of our research isn't actually like this. We spend less time riding horses, and we spend a lot more time doing things like this. So we actually do a lot of our research today studying plant evolution in the wet lab with lab coats on, studying the biology of living plants. We also spend a lot of time in dark rooms with expensive microscopes, beginning to understand key features of plants about their anatomy, their chemistry. And then the one bit of overlap you will find is we do spend a lot of time in fossil collections, a bit like some of the scenes from Indiana Jones, but the fossil collections that we visit are rarely places which they're not just warehouses full of large sealed wooden boxes with mysterious artifacts. These are typically well-ventilated, ideally storage locations of large numbers of fossil plants arranged and catalogued in order we can use for our research. So today, paleobotany really is an interdisciplinary science, and we use a variety of these different techniques, these modern different techniques, to really begin to tackle questions of plant evolution. In my research group, we call this interdisciplinary approach the molecular paleobotany approach, and therefore our group, the molecular paleobotany and evolution group. And we're really fortunate to be based up at the University of Edinburgh, and Edinburgh is a really ideal place to do this kind of research. We have abundant access to fossil plants at National Museum Scotland, to living plants at Royal Botanic Gardens, Edinburgh, and of course, where we're actually based in the School of Biology, is ideally situated for much of the lab work we do. And if you want to know more about the work we do, please follow this link to our lab homepage, where we give updates about our ongoing research. But there is one thing that unifies these three disparate strands, and that is that all of our different approaches often involve examining specimens under the microscope, and the specimens we typically examine under the microscope are known as thin sections. They're thin sections because they're often small and thin slices of plant that are so thin that you can actually shine light through them with a microscope and look at their internal structure. So, for example, we can take a variety of living species today, and if we want to understand their development, we'll take thin sections through their growing apices and study them under the microscope. If we want to know about gene expression, we take a similar approach. We might want to visualize whereabouts a gene is expressed within the anatomy of the plant. And what's amazing is in certain cases with fossils, we can also generate thin sections of fossils as well. And we can therefore examine them, such as this example here. We can examine these fossils like this under the microscope to learn about the plants in amazing detail as well. And so for us, thin sections are really informative. They give an amazing kind of window into huge amounts of diversity of plant life. And in particular, in the case of fossils, they give us a window into the past. And over the rest of this lecture, I'm going to be giving you examples about how we use these fossil thin sections. But really, the story of fossil thin sections starts where I'm based, in Edinburgh. And so I'm going to start off this lecture just by giving you a bit of context as to where these first fossil thin sections were created and how they've gone on to change the science of paleobotany. So our story really begins here in Edinburgh, and it starts life at a place called Craigleith, which was a sandstone quarry. Here's a photo of what Craigleith Quarry looked like from the 1880s, looking towards Edinburgh Castle and to Arthur Seat in the background. Now, at this stage, this was a quarry way on the outskirts of Edinburgh, but today this has been swallowed up and is now just a retail park. But what's important about this quarry is they were quarrying huge amounts of sandstone, and it was sandstone from this quarry which had been quarried there for 300 years, which gave rise to things like the Edinburgh Castle and the Edinburgh University Old College. And while workmen were quarrying in this site, they came across some quite remarkable plant fossils. So the first of these were discovered in the 1830s while they were quarrying, and they came across some enormous fossil tree trunks. And I really love these early etchings just illustrating just how big and impressive these tree trunks were. And when the workmen found them, they, as you can see, they excavated around them, recognizing them already to be really interesting and exciting objects. And they were reported in the national news at the time when they were found. And in fact, you may have actually seen one of these trees, because actually these two, there was a number of trees, but two of them in particular are arguably some of the most famous fossil plants, especially in the UK. And that's because these trees sit in front of, one of these trees sits in front of the Natural History Museum in London. So if you visit today, this has been there for a long time. This is the new garden outside the Natural History Museum in London with the new sculpture of Dippy here. And this is one of the trees that was actually discovered at Craigleith Quarry. The other one, for many, many years, has sat outside the Botanic Gardens, the Palm House, and is currently not on display because it's undergoing regeneration as part of the Biomes project. But if you've ever been to the gardens or you've ever been to the Natural History Museum, you may have seen these trees that originally came from the centre of Edinburgh. Now, what's important about these trees is they're amazing in terms of their size and their structure. But importantly, if you look closely at these trees, you begin to realize that they actually have some anatomical detail, some internal structural detail. And this was first recognized very early after they were first discovered. And in fact, this man, William Nicol, realized that this actually studying the internal structure of these trees could be really illuminating for learning more about fossils in general. And so what he was able to do was to take one of these, take one of the fossils, and then he cemented it to a large piece of glass. You can see this one here. He cemented it using Canada balsam, and then he was able to manually grind down the thickness of that fossil, gradually making it thinner and thinner until eventually light could actually be passed through it, and you can begin to see these structures here. And what's amazing is we actually still have a record of these early thin sections. This is in the Hooker collection at the British Geological Survey, and you can see it's from Craigleith Quarry, the Great Tree from 1831. So this was the first time that a fossil thin section had been made, and it was the start of what was to become a revolution in how we study fossil plants in general. And it's actually been at that stage, people had already recognized that this was going to be a really important mechanism for studying fossils. So rather than having to manually grind down the fossil yourself, which is a lot of work, it wasn't long before people developed more automated ways to do this. And I really like this. This is an example from Balfour's 1872 book when he's describing the innovation of designing a machine which is going to help make those thin sections for you. In this case, it's Mr. Bryson was able to produce this machine by putting a large cutting wheel onto a leaf and then being able to use that to both cut specimens and to then be able to polish them down from a much thinner part. And back in the 1850s, you could have bought one of these for yourself for roughly £10 if you happened to be part of the fact that it's got a touchscreen and a lot more safety features. It's actually very similar. You can see the example down here of the cutting wheel looks almost identical. And what we're using, if we're using the cutting wheel to cut these thin slices of rock. So this has really become one of the staples for the study of paleobotany, the ability to cut these very thin slices of rock, which can then be examined. But you might be wondering, okay, so what do we get out of doing this? Why bother to produce these? Why go to all the effort? And the answer becomes because we can learn a huge amount about the plants preserved as fossils by studying their anatomy. If we take the example of those wonderful trees, the cradleaf trees described in the 1830s. From the surface, as you can see here outside the gardens, there's not much you can tell. You can tell it was a pretty big tree. You can say it had a large tree trunk. However, look at the internal anatomy, and these are the original plates from those 1830 papers. We can see the individual cellular structure within the wood. Now, the cellular structure of wood is a diagnostic feature, and therefore allows us to much more confidently to be able to predict what type of actual wood or what type of tree this was. And so by doing this, back in the 1830s, they were able to recognize that these trees shared a large amount of similarities to modern conifers today. And so this was really the starting place of realizing that by studying thin sections like this, we could learn key facts of their anatomy. And with anatomy, we could do a whole host of things. We could think about the physiology. We could think about comparisons with living species as well. But fundamentally, what I like so much about these is that these really do look almost literally like a window into the past. The beauty of having it so thin that you can shine light through is really like a stained glass window where we can look into plant life in the past. And so really what I'm hoping to do for the rest of the lecture, after introducing you now to what these things are, is to demonstrate how, more than coming up to 200 years later, we're still using thin sections and we're still being able to learn a huge amount about the biology of plants from studying them. So as I said, I've told you already about what thin sections are and how they were created at this time early in Edinburgh. But I want to give you two examples from the research we've been doing in the lab about how we're still using thin sections. And so the first of these, we're going to be taking a bit of a geological field trip back in time, focusing on some fossil sites in Scotland. And our first stopping point is to focus in on the Devonian period. So when we're thinking about the Devonian, we need to start really picturing a very, very different world from the one we're in today. So what we're doing in terms of time is we're looking at about 400 million years ago. We're particularly interested in the early Devonian, roughly 410 million years ago. This is a paleocontinental reconstruction showing where the tectonic plates were at this time. So a vastly different world. In fact, the vast majority of the landmass appears to be was in the southern hemisphere. And the UK at that point was actually situated down here south of the equator. And it was situated south of the equator in this early Devonian world. And what we want to do now is to then go hunting for these rocks in the UK. There's actually a number of sites in the UK where you can find Devonian rocks, but we're very lucky in Scotland to have quite a few of these sites around, including some very famous ones I'm going to tell you about soon. So in particular, some of the most famous Devonian rocks come from quite an unusual place to be looking for rocks, a small village called Rhynie in Aberdeenshire. So here's just a map of the UK. We start exactly where I am in Edinburgh, and you drive about 200 kilometers north. You get to the Aberdeenshire countryside. Here's Aberdeen here. If you go inland from here into the rural heart of Aberdeenshire, you get to this small village of Rhynie. Now, if you visit Rhynie today, it's a tiny little village nestled in this agricultural land. As a geologist, I mean, frankly, it's not a great place to be looking for rocks. As you can see, there's no large exposures, there's no big limestone scarps, there's very little rocks to be found at all. So it seems like an unusual place to be incredibly famous amongst the paleobotany and the paleontology community. And even if you zoom in to where this yellow arrow is, which is the SSI, so the Site of Special Scientific Interest, you're always far more likely to find sheep grazing in this field than you are to find fossils on the surface. But the reason for that is this agricultural land here, this green field with the sheep grazing on it, actually hides below it quite a remarkable secret. And that secret is a series of rock beds that are only evident when we actually dig down below the surface. So the last time this happened was just over 20 years ago, back in 2003. This is the first time the last time they dug a trench in the field of Rhynie. And this is a wonderful image shared by Dr. Andy Ross of what that site was like. So they dug down through about 6 foot of soil and eventually began to hit these black layers. You can see them at the base of the trench here. And what we're finding here is an unusual type of rock. It's very hard, it's very glassy, and actually from the surface it doesn't look like that much. You can see it here. It doesn't look anything particularly that special. But amazingly, this rock actually holds just an amazing amount of information, which has been well studied now for 100 years and continues to reveal exciting findings about an ecosystem that lived there. And the reason for that is this rock preserves something very specific. It actually preserves evidence that in this site, which is now, as I said, nestled in kind of rural Aberdeenshire, was once a geothermal environment, much more similar to what we see in somewhere like Yellowstone National Park or Rotorua in New Zealand or even Iceland. There would have been exploding geysers and bubbling hot springs and hot pools. And this is what we find evidence of in the Riney Chert. So to zoom into this in a bit more detail and to begin to understand it, we can look in detail at somewhere like Rotorua. If you go to Rotorua and you have a look at these bubbling, you look around the exploding geysers and the bubbling hot springs, you actually can see that there's still some life hanging on and growing around these environments. You can see some plants growing towards the edge of these bubbling hot springs. And at the same point, what you can also see is surrounding these bubbling hot springs, there seems to be this kind of layer of white. You can see the white texture around there. So let's zoom in a little bit further. And if you zoom in on the very edge of one of those bubbling hot springs, again, you see that you're actually getting a buildup of this white sediment over basically everything it comes into contact with. And what this is that in the water, this hot and geothermal water is laden with silica. And as that water bubbles out, as it overflows, as it floods and comes into contact with almost anything it touches, it then deposits that silica in the form of what we call a sinter. So this is what this white kind of covering you're seeing on everything there is. And what's amazing about silica is effectively the process we're looking at there is gradually from a sinter, it turns to opal. And then effectively what we're looking at is anything it comes into contact with is being gradually turned into glass. So the silica being deposited, this is what the main thing in glass is silica. And again, we're getting that same thing happening here. So let's just take this example of a pine cone. This pine cone has fallen into this environment, and gradually you can see that it's getting silica built up on its outside. Not only is the silica building up on the outside and entombing it, but it's actually silica is able to permeate within these specimens as well and actually preserve their internal structure. And so this pine cone is well on its way to becoming a fossil. And in the case of the Riney Chert, something almost identical must have been happening in the past in Riney roughly 407 million years ago. Those plants and organisms, that entire ecosystem that grew around these hot springs was gradually covered in silica and entombed in glass. And that made after millions of years, what we're left with is that rock we saw in the bottom of those trenches, which today is in many museum cabinets around the world as an incredibly famous deposit, known as the Riney Chert. So the chert is the type of rock, and it's named Riney after where it was found. So today, again, if you go to a museum collection, such as in National Museum Scotland, you can pull out drawers and drawers of this rock, which, again, from the surface doesn't look particularly exciting. But let's take that same approach that they did with the cradle leaf trees. Let's take these specimens, let's cut them, let's generate these thin sections. And when we get them, we get the structures like this. And what's interesting is even from here, you can begin to see there are structures within this thin section. We can see kind of circular structures, we can see some dark areas. And what's incredible is that these are actually the plants preserved and that ecosystem preserved within glass. And so when we cut it so thin, these are just excellent to examine under the microscope. So we take one of the thin sections and examine it under the microscope. What we're able to find is that the plants preserved within it are preserved with exceptional levels of preservation. This is a truly remarkable example of a transverse section through the stem of a plant. That plant is called Rhynia, and those stems are only a few millimeters in diameter. And within the stem, you can actually see all the individual cells, individual tissues. We can see an epidermis around the outside. We can see these cortex, these very round cells. And then we move towards the vascular tissue with the water conducting xylem at the very center. So what's amazing is these plants are preserved in a truly remarkable level of detail at a time when most fossils we have barely preserve anything approaching this level of detail. And so from studying the Riney Chert, we built up this complex picture of an amazing ecosystem that was living in the Devonian period surrounding these hot springs. So there's been about 100 years of research on the Riney Chert. And from that, we've been able to conclude not just about the plants, but actually about the plants that the animals they were interacting with and the fungi as well. So we have the earliest evidence of things like AM symbiosis, the symbiotic relationship between plants and fungi. We have stages in the plant life cycle which are not preserved anywhere else, and a remarkable diversity of early animals as well. And so taking all this together, what Riney is giving us is our earliest preserved terrestrial ecosystem, and therefore has been hugely illuminating for trying to picture what life was like on land in the early Devonian. So from the Riney chart, we've learned a huge number of things. We have a huge amount of evidence about early animals, including the earliest insects. We have the earliest evidence of fungal symbiosis with plants, and therefore demonstrating very clearly that the early evolution of plants on land was not done in isolation, but was really always and has been done hand in hand with fungi. There's a huge amount about microbial life, and not to mention a host of things to do with the evolution of plants, such as the origin of roots and of leaves. But our most recent investigation into Riney was actually onto something quite different. And that was our focus on a Devonian enigma known as prototaxites. So when I like to picture a Devonian ecosystem, and the story I've told you so far is really summed up in this piece of art by Richard Bisley, who does some really quite amazing paleo reconstructions about what life was like in the past. This is a kind of Devonian ecosystem. It's plant life, it's on the margins of water, and a covering of plant biology which would barely even get to your shins, so only a few centimeters in size. This is the main picture of what we picture the early Devonian world looked like. However, there is an elephant in the room, and it is a literal elephant, because actually another reconstruction from Bisley shows the early Devonian to look much more like this. An alien landscape stranger than science fiction. These literal elephants in the room are these huge columnar structures known as prototaxites. Now, prototaxites really is the star of the show. It was recently the star of BBC Earth, these towering colossal structures. They definitely were the star of the show during this early period of terrestrialization of plants as plants became more complex. We have clear evidence of these fossils from around 425 to 365 million years ago. So during their main dominance was during the Devonian period. And then at that point, they went extinct. And it's been an enigma about how best to interpret these plants and what to compare them with, and really what they are. And really, the story goes back to the very origin of these first early prototaxites fossils. Starting with the name. So the name from prototaxites they were first found in 1857, and then they were named in 1859. And they were called prototaxites because when they were first found by Dawson, they were interpreted to be the rotting wood of a taxus tree, of a yew tree. And so he named it prototaxites. And you can see why. This is one of his illustrations from 1869. You can see it has these kind of tree ring-like structures, and you can see why he might have thought it was a tree. Not to mention these kind of giant size of these organisms, where we know that their fossils, you can see a huge example of one here, and another example of these here, illustrating that these organisms could get to about 8 meters tall and 1 meter wide. And then we're presented with these fossils, you would therefore assume anything that was giant and tree-like was most likely to be a tree. However, this kicked off a lively and very much ongoing debate about what on earth these organisms are and how do we rule in and rule out the different types of organisms. So during its long history of being studied, prototaxites has been interpreted as, and also not limited to this list, but has been interpreted as a brown algae, a red algae, a land plant, a lichen, and a variety of different fungi. And with each one of these different interpretations comes an amazing new paleo reconstruction, where these organisms, giant columnar tree trunk-like structures dominating the ecosystem, where they in fact rolled up mats of vegetation which have simply rolled down slopes into water, or instead where they giant creeping rhizomorphs, these kind of structures similar to what we find in some fungi today. And really, the reason why there is so much uncertainty and so much disagreement between scientists comes down to the study of prototaxites with thin sections under the microscope. So when you investigate prototaxites, when you make those thin sections through those giant tree-like structures, what you find is a quite unusual internal anatomy. The internal anatomy seems to be composed largely of tubes. So if you cut a longitudinal section through prototaxites along its long axis, you find them all arranged a bit like these large spaghetti tubes. Whereas if you look at them from the transverse section, you see all the circular tubes aligned in order. And when you keep magnifying it, you realize that this isn't just a single type of tubes. This seems to be a network of tubes within tubes within tubes. So we've got these large ones, and they're surrounded by these smaller ones. And so basically what we can see in Prototaxites is its overall organization seems to lack obvious tissues and organs like we see in other organisms, and instead just seems to be an interlaced mass of tubes of different sizes. And so based on this, many of those early hypotheses for what it could be have been largely ruled out. But there's been a growing consensus over at least the last 30 years of two main take-home messages. The first is obviously that there is nothing alive today which truly is analogous or that's similar to Prototaxites. So with this in mind, in some ways it could almost feel like a futile hope to actually be able to assign it to a modern group. However, it's still a really great goal to try and understand where this lineage came from and whether it has any close relatives alive today. And actually, the second major thing that's come out is, first of all, that we know that there's nothing truly analogous today. And then the prediction, therefore, has kind of divided into two. One suggests that it's most likely to be some form of fungus. And the other major hypothesis is that this organism just simply has no living relatives at all today. And we've just, in the sense that we just can't assign it to any living group alive today. So what this means is that many of the biggest questions still remain entirely unanswered about Prototaxites. There's been a lot of thought that it's likely to be a fungus, but with that in mind, we still don't really know what the organism would look like if you were standing next to it. We don't know why these organisms were just so different. Why be enormous in size when the rest of the terrestrial vegetation was so small? And at that same point, why did they go extinct? And why did that extinction correlate with the origin of land plants becoming trees? And then can we confidently assign it to a modern group of fungi? If we do think it's likely to be a fungi, can we assign it to a modern group? And this is where the famous fossil site, the Rhynie chert, comes into its own. And the reason for that is the Rhynie chert has evidence of, as I've mentioned already, has evidence of plants, of fungi, of animals, of a huge variety of different organisms, all in one geological setting. So it's an ideal place to build comparisons. So what we really wanted then was to actually find evidence of some fossils, of some fossil Prototaxites in Rhynie. And this is work that Laura Cooper, a PhD student in the lab, has really led. So what we're able to do was to hunt through blocks of fossils of the Rhynie chert, and we came across this one here, which looked just a bit atypical because it had this large grey mass in this region here. But it wasn't until we cut that specimen down that we realized that within there was this large spotty structure. You can see the kind of dotted structure indicative of Prototaxites, and in particular this one species, Prototaxites tatii, from the Rhynie chert. So at that point, we realized we had a new specimen, and that gave us the option of producing new thin sections and therefore being able to use a whole suite of new imaging and analytical techniques on Prototaxites, all in this setting, the Rhynie chert, where we have a variety of other species growing as well. So what Laura was able to do is to generate these thin sections and then to take them and examine them with the most state-of-the-art microscopes that we could get our hands on. And so Laura did some amazing imaging using confocal laser scanning microscopy. Now, this is a technique which is really the workhorse of much of molecular biology. You use lasers to excite your thing of interest. In molecular biology, that might be a particular protein or a particular gene. But in our case, what we can do is we can use the lasers simply to excite the material, the biological material still in the fossil. And that's able to autofluoresce and give us a signal. So taking that thin section where we weren't able to see a huge amount of information and look at the tiny, minute structure now under the confocal, Laura could begin to actually identify these different tube types, tiny, tiny structures within these tubes, such as evidence of septi amongst some of the tubes and a variety of the different structures. And in particular, we were most drawn to those spotty regions that I showed on the last slide. From the surface, these are indicative of having this spotty-like structure. And when you look at these in light microscopy images, they appear to have a shape such as a dot. So you've got lots of tubes kind of heading into this main region of a dot. But it's always been a little bit uncertain about what these structures are, or actually about what they mean for that overall network of tubes we find outside. So outside these tube networks, as I said, we can document these different types of tubes, these larger ones, these smaller ones, but how does that relate to the spots in general? So what Laura could do was actually to take these images under the confocal, and what's amazing about those is you're able to collect data not just from one plane, but actually to build up a 3D reconstruction about what those tubes look like. And by doing that, you can actually visualize the complexity of the branching in one of these tube points. And this is what this 3D model is here. So these, in a single plane, you can see these structures shown in blue. These are actually on the surface. And then Laura could actually connect up all the tubes that connected to those tubes actually went down in this 3D model. And the result from that was a hugely complex network of tubes all amalgamated together, as you can see there. And what's exciting for us about this is two main things. The first is that we're struck by the connections between these disparate tubes and their hugely high density. This high density appears to have a hugely high surface area. And if we saw one of these if we have any analogies for these in other organisms, these are normally structures involved in mineral or nutrient exchange, anything which involves a lot of exchange of nutrients with this giant surface area. So this was beginning to tell us a bit more about physiologically what these structures might be. But importantly for our interpretation of fungi, what was particularly exciting is that this level of organization, where the tiny tubes connect to larger tubes all in one isolated place inside a spot region like this, is actually really unusual and is not something we could find any evidence for in any living fungi today. So what this told us is, in fact, based on anatomy of these fossils, their anatomy doesn't actually support the fungal affinity. Yes, they have tubes, but the tubes are organized in a very different way and therefore doesn't support the fact that they're similar to modern fungi. So this was the first thing we were able to leverage with these new thin sections. The second thing we're able to leverage was actually to look at the chemistry and the chemical fingerprint. So this started a really fruitful collaboration with Quentin Long and Sean McMahon over in the School of Physics and Astronomy. So what they'd been working on the Rhynie chert previously and are able to identify that there was actually, within the fossils within the Rhynie chert, there was a chemical fingerprint for different organisms. So that when you investigated them using Fourier transformed infrared spectroscopy, you can see here, you can actually, from that, you can actually distinguish between the different groups. We could get the plants over here, the fungi over here, the arthropods. And this allowed us an analytical tool for distinguishing between different fossil types. So what we wanted to do was, in teaming up with we're able to stick them through the software, and in this case what we're able to find was to identify that Prototaxites, this unusual organism, was sitting all the way out here on its own, completely different to the other organisms in the Rhynie chert. So very different to the plants, to the arthropods, to the fungi, and even to the cyanobacteria that we know are preserved in the Rhynie chert. So this is by combining the evidence from the FTIR approach, and even by looking at the Prototaxites in amazing detail using synchrotron FTIR as well. So taking this all together, what this began to tell us is a story where up here in our kind of chemical space, we could find the fossilization products of phenolic compounds, such as things a bit more like lignin that you'd find amongst, you find in plants. Whereas down here, we find the fossilization products associated with chitin. So what's interesting about chitin is the main polymer that we find in things like arthropods, and it's also how fungi, one of the key components within fungi. And so if Prototaxites was a fungus, we would really expect it to have fossilization products which looked similar to the fossilization products of chitin. And what's extremely exciting for us is in the Rhynie chert, we have these clear fungi, such as these ones here, and we're able to find that they do have evidence of the fossilization products we'd expect to find, whereas we don't find those in Prototaxites. So taking these both together, what we could then conclude is that both the anatomy and the chemistry of our new fossils didn't seem to support a fungal affinity. Instead, this sent us back to the drawing board. Because basically, if our new evidence suggests that these were not fungi, then the question becomes, and circles back around to the early question that was being asked all the way back in the 1870s, which is what to do with Prototaxites. And I really like the fact that this has actually been posed as a question in a paper by Nelson and Boyce from 2022. It's this ongoing debate about what to do with this organism because it's so difficult to assign it to any known group. So many people have led to the conclusion that it likely had affinities with fungi. But our new study actually said the opposite, that both anatomy and chemistry seem to support the fact that this wasn't a fungus, or that didn't seem to share similarities with any known fungi we knew today. So with that in mind, we opted instead for a kind of a more conservative approach, and we acknowledged the fact that really, if we can't be confident it's a fungi, why try to shoehorn it into being a fungus? Maybe it's better just to take a step back and say, actually, this is an organism we truly don't understand, and we don't know anything about it, in the sense we can't assign it to a modern lineage. So we can assume that it was likely a eukaryote, given its type of complexity. We knew it was growing on land, so it's a complex multicellular eukaryote, but actually being able to assign it to any known group we think is not possible at the time, and therefore we interpret this as an organism which likely its entire lineage went extinct, and that we are still currently unsure about what it is. However, what we definitely can conclude is that it was a key taxa within the Rhynie ecosystem. And so, teaming up with the artist Matt Humpage, we were able to reconstruct what that Rhynie ecosystem might have looked like in 3D. Up to now, most of our work had been focused on understanding the plants you can see here. But amongst these plants, there must have also been these prototaxites organisms. Whatever they were in some ways maybe being analogous to fungi within the environment, but they certainly weren't fungi in the sense that they didn't share anatomy or chemistry with the comparable fungi that we know from there at the time. So this was just a great example to show you how a new finding of a fossil and making thin sections in the same way that we've been making them for 150 years, but using those thin sections with a whole range of new state-of-the-art imaging and chemical techniques, we can learn a lot more about an enigmatic organism, even if the overall conclusion from that is to say that we are certain that we are uncertain about its affinity and that we still don't know what it is. Okay, and then for the final part of the talk, we're going to be jumping forward in time a little bit, and we're going to move to focus in on how we're using these same techniques, this time to investigate a fossil from the Jurassic of Scotland. So on our little field trip, we've moved our way up from Edinburgh. We drove the 200 kilometers from Edinburgh to Rhynie, and then if we keep going further past Inverness onto the northeast coast, we get to the small village of Helmsdale. Now, geologically speaking, Helmsdale is a pretty interesting area, and you can see this by looking at the geological map of that region. Now, the different geological strata are coloured in different patterns, and what you can clearly see about this area of Scotland is what you're looking at is something that looks much more akin to a piece of modern art than a very regular stratigraphy. So there's a huge number of different rocks all in one place. And if we zoom in particular and actually make the comparisons with where we've been already with Rhynie, we see that there's a variety of rocks all kind of smushed up together. So if we start over here, you can see the main sort of pink color. These rocks are actually from the Silurian period. So this is marked on the stratigraphy here, around 430 million years, so older than Rhynie, where we've been already. Jump forward a little bit further, if we're looking down at the south of Helmsdale, what we see is rocks from the Devonian period, but this time about 390 million years. So for moving from the rocks number 1 to rocks number 2, we're actually spanning the period of time when the Rhynie chert was formed itself. But next to those, you can see a large band of rock, which you might therefore assume to be somewhere in the sequence, somewhere maybe linking these Devonian rocks. But actually, when you look at it in more detail, what you find is in fact that these rocks here actually preserve evidence of the Jurassic period for Scotland 150 million years ago. So there's a huge amount of geological time separating the rocks on this thin sliver from their surrounding rocks. Now, let's take a jump into the Jurassic. So we can leave the world of the Devonian behind, where the UK was based in the southern hemisphere. In that time, it has gradually transitioned north. An enormous supercontinent was formed, Pangaea, and now we're in the Jurassic period. Roughly 150 million years ago, that supercontinent is fracturing and breaking apart. Now, as it's fracturing and breaking apart, the UK, which is situated around here now, is being surrounded by shallow seas. And this is a period of time of high sea level of a warm world in the Jurassic. And so these shallow warm seas gave rise to an amazing diversity of animals on land. And actually, for most people, if you think about the Jurassic and the Jurassic rocks of the UK, the first thing that's coming to mind is the famous English Jurassic coast, where you can find abundant ammonites, such as this beautiful example of a large ammonite with all the small ammonites within it. However, within Scotland, we actually have some evidence of terrestrial organisms. So not only do we have evidence of life in the water, but actually clear evidence of life on land. And I have some really brilliant colleagues here in Edinburgh who've been doing some excellent work on those amazing ecosystems. So this includes finding evidence of giant dinosaur footprints and evidence even of our early ancestors, early mammals, making life in these early life during the Jurassic. But what's interesting in these cases is most of these studies have focused on either the kind of footprints or isolated remains of the animals like dinosaurs, or the small fossils representing the skulls. And in actual fact, we're still quite uncertain about what environments these organisms lived in. And that's what we were really interested to try and find out. So to do that, we went to we went to Helmsdale, and when we're in Helmsdale, the thing that we're hunting is actually depicted in this image with the dinosaurs here. It's these bits of wood, and actually there's large amounts of this wood which is actually found alongside dinosaurs. And actually, when coming to Edinburgh, it was Steve who told me that often when they're out hunting for dinosaurs and dinosaur fossils, they often come across bits of fossilized wood, and wondering whether there's some interesting research that could be done on those. So this is the motivation to go to Helmsdale, to go hunting along that Jurassic coastline, the Scottish Jurassic coastline you can see here, in search of evidence of fossilized wood. And what was kind of remarkable, and I really didn't expect this, was just the amount of fossil wood that you could find. So by walking on the coast, you can actually see quite a lot of evidence of fossil wood. It's so easy to walk past because it looks so similar to the surrounding stones, but once you get your eye in and start spotting it, you start to recognize the key features of the fossil wood. You can almost begin to see little tree rings in this. This looks almost identical to the charcoal you might use on your barbecue today, and even some larger bits of fossil wood as well. And so, as I've told you so far already in this talk, the motivation then is not just finding these, but actually creating new thin sections from these. And these thin sections offer the potential to shine light onto these early ecosystems. And so when you create thin sections of this wood, you get a really, really incredible insight into life during the Jurassic period. In this case, this is a wonderful example from the Hunterian Museum, just showing you the details, the anatomical details preserved in a piece of fossil wood. I'm always struck by just how amazing this is and how we can see the tree rings, and therefore how we can count up the tree rings of a plant that lived in the past. So in this case, we can actually follow and count the tree rings in a tree which grew over 150 million years ago. So it's such a kind of tangible thing to think about 15 years in the life of an organism that lived 150 million years ago. And during that time, there were years like this one that looked like year 11, where the growth rings are very large. It must have been a good year for being able to grow, whereas other years we find much smaller, narrower growth rings, maybe a more stressful year. So this gives us a real insight into what these ecosystems must have looked like. And from studying that wood anatomy as well, we can learn that the wood we're looking at is very similar to conifer wood today. And another great example from Bisley art, we can see what these kind of conifer trees might have looked like during the Jurassic. However, that in itself doesn't necessarily tell us about the complexity of the ecosystem. It just tells us more about one plant and the life of that one plant during a small space of time. But it's while looking at these fossils that we came across what to the untrained eye might have been actually a rather boring, rather weathered specimen, this example here. It has a kind of mottled texture, you can see that there, and therefore in many cases wouldn't be one you'd want to pick up and make those beautiful thin sections of. However, very, very lucky here in Edinburgh to have a brilliant postdoc, Anahu Sagasti, who actually brings a lot of experience from studying Jurassic wood, in particular from Patagonia. And so when showing this specimen to Anahu, she was able to look at it and be able to recognize that actually we're looking at something very interesting. This mottled texture which makes it less exciting for studying maybe the tree rings, actually gives rise to some really interesting biology. And as you can see, we need to make new thin sections and examine it under the microscope. I went and who was investigating it under the microscope, and you can see this on the monitor here. You can see that there weren't the clear evidence of the tree rings. Instead, the wood seemed to be fractured and broken up. And from looking at that in more detail, what we realized is what we're looking at is a piece of wood that had actually rotten. It was rotten wood, it was falling apart, and it had a variety not just of plants, but of other organisms in there as well. Now you may think, what's the interest in rotten wood from the past? Well, one of the best ways to think about that is to think about the role of rotten wood in a modern wood today, a modern ecosystem today. And rotten wood is really quite amazing. It can offer a home for organisms to live in. It can be a substrate for things to eat, to grow on, to live in, to have part of their life cycle, for plants to grow on, for fungi, animals, and a whole range of other organisms thrive and survive off fallen wood in forests. And so that made it very interesting for us as an opportunity to investigate the wood structure in a lot more detail. So what we wanted to do was to investigate it and see what evidence we have of these complex processes of decay and rot in a Jurassic forest 150 million years ago. So the first thing we noticed is, as I've already mentioned, when we looked at the thin sections, there was no evidence of those lovely pristine growth rings I told you about previously. Instead, when we look closely, the cells of the wood, you can see here, appear to be breaking down, actually breaking down into these pockets, these holes or pockets. And when we zoomed in more detail, there's actually a really diagnostic pattern where you could see evidence that the cell walls, which are normally quite thick and dark, were gradually being eroded through into these whiter regions and eventually into holes. Now, this is completely diagnostic of the action of fungi, and in particular, a type of fungi known as white rot fungi. And so we then looked in a lot more detail. We zoomed in further to look for evidence, if we could find evidence of fungi, and we found clear evidence of fungal hyphae within the cells and also fungal spores as well. And so what this seemed to tell us is that one of the first processes that was happening to this wood is that as it was being broken down, it was being colonized by fungi, and they were actually able to physically break down the specimen in the same way that many living species we see white rot doing the same thing today. So the first evidence of decay seemed to be by fungi, and then after the fungi has begun to soften the wood and make it more palatable for other organisms, the next big thing we could see was evidence of large galleries. So if we're looking at this large thin section here, we can see the gradual shape and the structure of those tree rings, the radial pattern moving outwards here. However, interspersing these big areas in red, another one here, which just seem to be full of loose tissue, woody tissue, which we call frass. And when we look in particular around the borders of these, we find this kind of rough outline. And this is completely indicative of the feeding strategies we find of the larvae of beetles today. And so what we're able to see is actually not only has there been evidence of the fungi, but actually this appears to be clear evidence that animals are actually are boring their way through this wood as it was rotting. And it's not just these larger larvae we have evidence of. When we zoom in more detail, we find these smaller holes, such as this kind of circular outline here and this one here. And these are filled with these small little circles, these little pellets. And what these are actually fossil coprolites or fecal pellets of tiny animals, very, very similar to what we find today in orchid mites. And so you can see them, you can see that this is clear evidence that the mites are actually getting in and they're burrowing their way through, and they're leaving the direct evidence of where they've been in the form of their fossil coprolites, you can see here. So incredibly, we have evidence not just of one kingdom of the fungi, but the animals in this piece of decayed wood as well. And then finally, amongst these, we found something that as plant scientists was particularly interesting. We found clear evidence that within this fossil wood, there seemed to be rootlets. So small roots or small tree roots, plant roots, sorry, are growing in this wood as well. When we look at it in the transverse section, we can see the diagnostic features of the outline, and we can see the water conducting xylem at the center of these. And this is completely indicative of the fact that a plant has actually been using this wood as a substrate for itself, has actually been growing its roots through it. And actually, although we can't be certain about what species it is, we think it's reasonably likely that these roots may have been from another conifer. So potentially a conifer growing down into this decaying wood. Now, the reason why finding these roots are so important is it gives us a novel insight into a really, really important ecological strategy. And that strategy is known as the nurse log strategy. So one of the reasons why keeping fallen wood on the forest floor is so important is that these bits of fallen wood and decaying wood can actually act as an ideal nursery for new plants to get started and for the process of forest regeneration. So actually trying to germinate and grow on the forest floor can be really challenging. And so that's why in many cases, seeds, when they fall, they can actually be very fortunate if they've landed in some decaying wood like this. And this offers an ideal place for the seeds to germinate, for their roots to go down into environments that are not often too waterlogged. And there's even evidence that actually this can be really beneficial for producing, for really getting established. And we can see these in modern ecosystems today. And in fact, amongst conifer forests today, we can find evidence that in a conifer forest, often up to 90% of trees growing in the forest might have in fact germinated on a fallen tree like this. So these structures are vitally important for the ecological succession of forests, and we appear to be having evidence of that present in the Jurassic. And so what we're able to recognize is there's been a very, very close evolutionary link between the evolution of fungi, which break down wood, and actually the new growth and regeneration of a forest itself. So taking all these lines together from this amazingly small fossil, which could have been so easily overlooked on the beach, we actually have a really complex ecosystem preserved there. We have evidence of fungi, we have evidence of at least 2 different types of animals, and even evidence that this bit of wood is being used as the structure for actually plants to get a footing itself. And therefore, this wood gives a real insight into the ecosystems that underpinned the amazing dinosaurs and the mammals that we're familiar with from the Jurassic as well. So with that just draws me to the very end of the talk. My aim for the talk today was to convince you that fossil thin sections can provide a unique window into prehistoric ecosystems. First telling you the story of their history and then how we're still using them today in the lab. So if you've been interested in this, please do follow and get in contact to learn more about the work we do at the University of Edinburgh in the Institute of Molecular Plant Sciences. We have a really active outreach team describing some of the ongoing work we do. And with that, I just need to say a huge thank you to my funders. This wouldn't be possible without their help and support, and to my amazing group. And with that, I'm very, very happy to answer any questions. Thank you very much.
Thanks ever so much, Sandy. That was a really fascinating talk, and I'm sure there'll be a lot of enthusiasm for questions. There are quite a few already in the Q&A, and so I'll kick off with the first, and it relates to mycorrhizas. What is the earliest association between plants and fungi that has been found? From Roger.
Thank you, Roger. That's a brilliant question. And actually, the earliest evidence is it's a very nice question in the sense that the earliest direct evidence comes from the Rhynie chert. So the Rhynie chert fossils where we found those, the prototaxites, also hold the earliest evidence of AM symbiosis between plants and fungi. Now, what's different about the AM symbiosis we see there is today that symbiosis is mainly specialized in just the rooting systems of the plants. Whereas in the Rhynie chert, the plants actually are at a stage before they have really clear, distinct roots and shoots. And so we're actually seeing evidence of that fungal symbiosis through quite a large portion of the stems, rather than just being isolated into these things. So it's the earliest evidence we have, and it's obviously the starting point of these much more complex interactions and specialization that came later.
Yeah, thanks, Sandy. Yeah, there's some beautiful images of those associations that are available if you search for them. So now one relating to that enigmatic prototaxites. has Prototaxites ever been linked to a terrestrial, to a bacterial deposit, or perhaps less likely cacti?
So both of those are interesting ideas. We'll deal with the bacterial one first. So in general, the level of complexity seems to indicate that it was a large multicellular organism. And that kind of makes us suggest that it was likely a eukaryote rather than a kind of prokaryotic organism. But it's a really good point because you can imagine you can get kind of bacterial mats, and there's been a few hypotheses about it potentially being a mat forming organism, which is then kind of being rolled up. But in general, the level of organization we see, and especially when you find them in the larger chunks, we find would be really unlikely to be that. And then for the second point about cacti, interesting, but cacti are much, much more recent. These are typically flowering plants that we just wouldn't expect in the fossil record at that point. This is around the time where most of the plant life are these kind of early vascular plants like lycophytes, and we're getting the split of ferns and gymnosperms. So I think there's been less comparisons, less comparisons with cacti. But again, in terms of an overall large conical structure, then maybe there are some physiological or kind of overall morphological comparisons that could be done.
Yeah. Some analogies, perhaps.
Yeah.
Yeah. And then from BM, how confident are you on the growth habit of Prototaxites? If plants were relatively short, why did it grow so tall?
A wonderful question. So we are confident that these things got large. The examples I showed of some of the biggest specimens, we can clearly see that these things could be on meter scale organisms. So we knew some of them could be absolutely enormous. However, I think within that, there's actually also a large number of much smaller examples as well. So the example we're looking at from the Rhynie chert is actually much, much smaller as well. So maybe there might already been a huge diversity within them, within their sort of size and scales. In terms of why they got so tall, that's always a wonderful question. I mean, the most obvious one that comes up through comparisons with other organisms is the potential benefits of having a huge structure like that for reproducing via something like spores. However, the evidence for that is currently lacking. So what we need is more fossils, and ideally we'd be hunting for fossils of fertile portions. And you can imagine it would be very illuminating if we found on a giant trunk that there was evidence of fertile portions on there. But until then, not confident at all is the answer, I think.
So just following on from that Sandy, have fossils of Prototaxites or things like that been discovered in other deposits that are not Rhynie chert or Devonian? You showed those Silurian deposits next to the Jurassic ones on the coast. Is there anything in the Silurian deposits?
Yeah, there certainly are. There's some fragments, and Professor Dan Edwards has done some really lovely work on the kind of latest Silurian into the Devonian as well, and the early Devonian with charcoalified fragments as well. So they've been around for quite a long time. They're found throughout the world in the Devonian, and there were likely evidence that is there in the Silurian. The issue with the Silurian, if we have an enormous rock record bias where we just don't have many rocks from the time period. So it's much easier to look and identify these things in the Devonian where we've got many more rocks, and when we have more rocks, we find more specimens. And so I think continued looking in the Silurian will be really useful. There's also a few earlier fossils which are very tentatively. Some people have interpreted as being maybe earlier records of Prototaxites as well. So there's other enigmatic tubular things going back into the rock record. So joining these together will be a really nice thing.
Thank you. Now from Carolyn, do you have a library of images of cycads, araucaria, etc.? And can you use AI to help you decide what a fossil structure matches today?
So that's a really interesting point. So we don't have a specific fossil data set of those particular groups. However, lots of that information does exist. It exists in collections, and I think that would be a really valuable way forward. So there's a number of these big digitization projects in a variety of things. I think herbarium specimens in botanic gardens are one of the greatest examples of this, where digitizing herbarium specimens of plants have then been used for a whole variety of other things. And I can imagine similar things being used for fossils. And I think the more images we can have of fossils, the more we could use things like AI to help train, maybe, and potentially just ask questions that were never possible before. So we haven't started doing this yet, but it's something we think about a lot. And I think the only issue we're at the moment is many of the fossils we look at are often of fossil soils or fossil ecosystems, which are they're inherently very complex, and they're the kind of things that our own eyes are very good at spotting, but actually for some of the machine learning algorithms, they still struggle quite a lot. But things are getting a lot better and more complex, and I think it's certainly something we need to look at for the future.
Yeah, interesting. AI in fossil research. Yes.
Yeah.
Good. And there's a question here from Alexis. Did you find any evidence in the Rhynie cherts of a polysporangiate plant, brackets, rhiniophyte-like plant, with a dominant gametophyte phase over the sporophytic one, like in bryophytes? Yeah, because there are not many fossil bryophytes around, or anything that we can
No,
see that is a bryophyte. So that's a really good question.
yeah, absolutely brilliant question. So for those who are less familiar, so plants alternate between these two parts of their life cycle, the haploid and the diploid phase. And in most vascular plants, you're going to have your dominant sporophyte phase is what we have. The Rhynie is exceptional because we do have evidence of both phases of the life cycle of these of the plants, which is something we almost never have in the fossil record.
Mm.
And when we find that, we get a very unusual pattern in that we find a level of complexity between both generations, which is actually comparatively similar to each other. So this has been termed as approaching an isomorphic generation. There are certainly differences between them, but it's a level of complexity and similarity which we don't see in any living groups today. So we have polysporangiophytes, where we have things like stomata and vascular tissue in both sporophytes and gametophytes. But specifically to answer your question, we have no evidence of a bryophyte-like life cycle in the Rhynie or from any of these early fossil sites. So we don't have anything that we can confidently say we have a dominant gametophyte. And this builds into this large enigma about where the bryophytes came from and how, and why their fossil record is quite so lacking.
Okay, well, that's a good one to, I think, end on, because we don't have any more questions. I'll just move over to the chat.
I was going to say, Simon, that there are two hiding in the chat, one from Paul Kenrick and one from Sue Burge.
Okay, so I'm just looking. Yeah, here's the one from Paul at the Natural History Museum, of course. Prototaxites is such an amazing and enigmatic organism. If it's not a fungus or a plant, is there any evidence of how it lived in terms of sourcing nutrition, energy to build such a huge structure?
Yeah, great question, Paul. And yeah, thank you very much for coming on to talk. I think there'll be other people on this call who will be looking forward to your talk as the next in the series. So interesting. So I think based on the evidence from mainly based on Kevin Boyce's work on the isotopes, the fractionation the isotopic range we're seeing suggests that it was likely heterotrophic. That's always been the prediction. Based on the Boyce isotope work. And so with that in mind, I think that's still the dominant idea about what the in the sense that it's most similar to a fungi in terms of how it's making its living. I think the main thing we're trying to highlight is that we just think it's both anatomically and chemically distinct from fungi. So it could have well been a completely different branch of the tree, which is come onto land independently, has evolved multicellularity independently, but seems to be making its life as a heterotroph. One really interesting thing is to think whether there is any evidence about it being autotrophic and potentially being photosynthetic. But in general, most of the evidence that we find, especially with this kind of columnar structure, really speaks against the idea that the thing was adaptive for capturing light, given that almost every other lineage that is autotrophic and capturing light typically has flat planar structures for doing it. It just seems quite unusual that it's not. But yeah, it's a really, really great question, and one that I hope we can continue talking about more as well.
Yeah, it'd be great to get to the bottom of it one day, won't
Yeah,
it? Yeah. Maybe you'll do it, Sandy. And then the final question from one of our regular listeners, Sue Burge, who says she found the talk truly fascinating, and she wants to know who first discovered the Rhynie chert.
Yeah, a great question. So there's a lovely story about a person called William Mackey, who was a doctor in Elgin. So Elgin's a little bit north. If you basically go from Rhynie and go north to the coast. And he was a doctor at the time, and in his spare time, he was a geologist, and he was mapping in his spare time was going around the local area and mapping the area out, looking at the different types of rocks. And it said, and there's a beautiful paper which describes it, that says that while mapping the rocks around Rhynie, he noticed in a dry stone wall that there was evidence of a type of rock, a chert, which looked really unfamiliar to him. And he was able to make a thin sections from that, and from doing that, he recognized there was plant material in there. And so already with this is just incredible that he found it. And then what's even more strange is then he was then able to actually track down where it is, this tiny localized field, by in theory looking at the distribution of where he was finding more of these cherts. So it is remarkable that this was first of all ever found, and let alone in a story like that, because yeah, I think it's quite amazing, because it really is sandwiched between rocks of incredibly different ages. And it is just a tiny amount, but since its finding, it's been localized effectively to just a couple of fields which have been dug up, and all that information, all that we've learned, has come from what's excavated from there. So it's a beautiful story about the early work and early enthusiasts have then actually pioneered all this work that's come from mining.
Yes, an amazing place, amazing what's going on underground there. Yes. So on that note, thank you so much, Sandy. That's a great start to our paleobotanical themed series of autumn lectures. So thanks again, Sandy. Big virtual clap from everyone in the audience. And just to note, the next lecture will be in 2 weeks' time. So I'll look forward to seeing you all again there. So good night, and thank you all, and thank you, Sandy.
Thanks, everyone. Bye.
Bye.